friend of mine wrote this book. It's called The Culture of Make-Believe. And that's what we are. We don't know anything. We believe in, in all kinds of nonsense. And I think maybe that's deliberate. I mean, I don't know, but that's so science, so-called science is just full of nonsense. And if you actually dissect it, one of the things that I don't just stop at the viral theory. When you actually look at what we know of as the components of a cell, like ribosomes and endoplasmic reticulum, and that nerves have synapses, and there's cell membrane with receptors like the ACE2 receptor.
Oh, that's right, the receptor.
Sodium potassium pump.
Yeah.
All these things are make-believe. They don't exist. And the reason you know that is because when you look at a ribosome, every published picture of a ribosome is a perfect sphere, a circle. You can only see it in an electron microscope. Then you say, well, how did you get that picture? So they took a tissue and they removed it from its living matrix. That's the first thing. Then they grind it up in a blender, and then they freeze it, and then they stain it, and then they put glutaraldehyde and alcohol, and then they shoot an electron beam at it, and they have heavy metal stains, and then it looks like a circle. Now, if I said to you, so I want to know what your hand is made of and what it does. So here's what I'm going to do. I'm going to cut your hand off your body. I'm going to put it in a blender. I'm going to freeze it to 180 degrees below zero. I'm going to stain it with osmium and silver stains. And then I'm going to shoot a high intensity electron beam that gets rid of all the water. And then I'm going to tell you what your hand looks like. And all it looks like is a bunch of stain on a slide. And that's how they found synapses, receptors, endoplasmic reticulum, ribosomes, you name it, all this.
Really?
And if you if you go looking at I spent, so anyways, a ribosome is a perfect sphere. Now, what are the chances if it's a perfect circle on a two-dimensional slide, then it must have been a sphere in real life, right?
Okay, yeah.
Now, what are the chances you take an orange, right? That's a sphere, and you put it in a blender, and then every piece of that is going to be a perfect circle? Zero.
Very little, yeah.
So you can't. So considering that that's how you got that picture, you have to conclude that that's an artifact of the staining process. And in fact, a guy named Harold Hillman proved that those things we call ribosomes are gas bubbles that are formed when you stain and dyeing tissue.
Oh
and
my
they
gosh.
pick up silver stain, and that's what you're seeing. There's no such thing. Now, if there's no ribosome, there's nowhere for mRNA to be made into protein, and then the whole theory of genetics falls apart.
It does. I mean, everything falls apart. Everything, because I thought this was all mapped out by these guys, but the whole thing is falling apart. So the whole understanding of what makes up the human body, the human cells, is all somewhat confabulated, is what you're suggesting?
Totally fabricated, because of a lot of reasons. And if anybody is interested in this, they need to read a guy named Harold Hillman, who was the master at dissecting how they came up with the idea of synapses. So the whole theory of how a nerve works, right, is synapses. You have this nerve, and then it comes to a moat, and then there's a chemical released called a neurotransmitter, and it swims across the moat and attaches to a receptor on the other side, and then the next nerve goes, right? That's neurology 101.
Yeah, there's lots of graphics out there on the internet showing how this happens.
Graphics. Now, if you do a dissection of a nerve, you never see a synapse. Never.
Wow.
Now, how do you get to see a synapse? So you take the tissue and you desiccate it. You take the nerve and you dry it, and then it pulls apart. And then you stain it, and you get stain accumulating at the two ends. And that becomes your synapse.
Oh.
Now, if you don't believe me, go looking for an actual photograph of a synapse. And you won't, all you'll see is electron microscope pictures of dots. And you think,
dots.
what the hell? That's not a synapse. And then you'll see all these beautiful pictures, drawings, you know, models of so-called synapse. And they're like the drawings of COVID, right? Or SARS-CoV-2. You see these spiky balls and all this stuff. That's not a real photograph. That's somebody at an art
There's
contest.
a lot of CGI. Yeah.
Those are art contests. And there's no evidence of a spike protein or anything. This is just sequences found in the dead and dying tissue. And the same with this lab created virus. They say, oh, it's a mixture of HIV and XMRV and SARS. It's not, it's just a bunch of sequences in a computer that they put together and made this new computer model. There is never been found the entire sequence of the genome in any sample anywhere. And if somebody can prove me wrong, I will publicly admit it. And I know for a fact that it can't be done because every sequence is assembled, not sequenced. There's a difference. Assemble means you take all these Lego pieces and you make a castle in your mind.
Mm-hmm. Yeah, no, I think you've explained
reality.
It's not reality. I think you've explained it beautifully. And in fact, I've spoken to other doctors, you know, and have read some information as well. For instance, Dr. Young talks about how it's basically chemical and radiation poisoning that affects the human body and creates disease. And when it attacks the red blood cells, there's a kind of an explosion. And I think Dr. William Trebbing talks about it as well. You know, there's a bit of an explosion and there's a lot of waste matter pushed out of the cell and exosomes and stuff and repair proteins and that kind of thing, which has bits of DNA from the cell, is being mistaken as a virus by these people. Yeah.
These particles that they show you, these are just debris. That's what happens when you poison. Could be radiation or, you know, could be chemicals. It could be thoughts. It could be feelings. It could be trauma. It could be lots of things. And you poison yourself, or the worst of it is having misconception. If you believe in make-believe, you then break yourself down because that's not good for you. Eventually, you'll do stupid things like wearing a mask or injecting nanoparticles in you and stuff like that. So you poison yourself and then you break down, and they call those viruses.
And they call those viruses. But you know what you've just talked about, just talking about it across the spectrum over here is kind of major, because you're talking about how faulty beliefs and misconceptions inform science and biology in a very large way. And things like synapses have not actually been identified under electron microscopes. And so that's sort of mind-blowing, actually. Sorry?
They've never seen it at a light microscope. They say it's too small. And so then they have this thing, electron microscope, and they don't show you a synapse. They show you dots. And they say even DNA was found with the original picture of DNA as a double helix was they dehydrated a nucleus. They extracted it with phosphoric acid. Then they exposed it to 62 continuous hours of an X-ray, right? And then they show you a picture, like an X-ray picture, that looks like an X chromosome. And they say, see, that's the double helix.
Oh my.
You think, are you kidding? Like that's has nothing to do with what would be found in a living person.